Bar-Or. Thank you very much for doing this interview with us here at Electrums. And so you're currently doing some research funded by the MSID and the MS Scientific Research Foundation on B cells. What is it that you're looking at in particular, and how will this enhance the progressive MS field in understanding the mechanism of that form of the disease?
So, Angelica, you're referring to the team grant that we have from the Research Foundation of the MS Society. We call it the Canadian B Cells in MS team. It involves co-principal investigators Jen Gomerman from U of T and Alexandre Pratt from the University of Montreal. We're very excited about this grant that's been in place now for over a year, and recent results of the clinical trials targeting B cells in patients with MS has only fueled the fire of excitement further. We think that B cells are important in multiple sclerosis in different ways over the course of MS, and in different ways in different compartments of MS. Of course, the target in MS is the central nervous system, the spinal cord and the brain, but we understand that relapses occur because of immune dysregulation in the periphery, meaning outside of the central nervous system, and then cells that go to the central nervous system and contribute. And that's what we call the relapsing biology. Very, very good evidence that B cells are important in that biology, and hence removing B cells, as with ocrelizumab, results in far fewer new relapses. We also are very interested in the possibility that B cells that are in the central nervous system and seem to persist there over years and years in the same person may be contributing to the injury that is underlying progressive MS, non-relapsing progressive MS. That, of course, represents one of the biggest unmet needs. And how different B cells can do different things is very relevant from a treatment perspective. One of the main advances in the field of understanding B cells is that we used to think, and textbooks still tell us, that B cells exist to make antibodies, one of the main ways that a body can defend itself against infections. But B cells can actually, independent of making antibodies, we now understand also regulate immune responses. And they can either enhance responses or downregulate responses. So depending on the B cell, it may be either a good guy or a bad guy in the context of multiple sclerosis. And while removing B cells with anti-CD20 removes many, many B cells, and in balance is better in decreasing activity, we would like to know better which B cells are the good guys and bad guys. Could we selectively target in the future only the good guys, maybe allowing for treatment to be used again and again and again with a safer long-term profile and so on. So there's a tremendous excitement in the basic immunology field with respect to B cell biology, in the neurobiology and neuroimmunology field with respect to B cells within the central nervous system, and understanding the mechanisms that drive B cells in one direction or another, with a major focus on translating the observations from the clinic back to the research lab, looking at human materials, complementing with very focused questions in animal models. This is really what represents our team effort overall.
And can you tell us a little bit about the clinical trial that looked at ocrelizumab in primary progressive MS patients?
Sure. As you know, we've heard from the press release already that this is a positive trial, which represents the very first positive trial ever in PPMS through phase 3. So the first most important thing is that this is very exciting, I think, for people living with MS, in particular primary progressive MS. There's some very interesting questions that come along with these results, and we'll hear more about the results tomorrow. One is, what are the implications, if any, to people who have secondary progressive MS? And there are many folks out there who would be very keen to understand the answer. And also, what might we learn biologically based on these results? Because this particular intervention is looking at removing selectively B cells of the immune system from patients. And we have realized over the last few years that although MS was considered to be a condition where T cells of the immune system play a particularly important role, we now realize that B cells of the immune system appear to play a very important role, at least in part by contributing to abnormal responses of the T cells. So it's not one or the other, it's both in a kind of cooperative way, and if you break the cascade by either targeting the T cells or the B cells or both, you may be able to mediate the same kind of benefit. What's unique about this particular study is that it was done in a group of individuals who had clinically primary progressive MS, and the question that we are asking ourselves as a community, are the positive results, meaning that taking away B cells slows down the progression of disability in these people with primary progressive MS, is it a reflection that they have, in ways that we haven't appreciated, some biological relapses under the surface and that it's benefiting them on that basis, because we do know that this exact same medication is very effective at decreasing relapses of people with relapsing forms of MS. Or, and it's not mutually exclusive, it could be a combination of the two, but is it the case that B cells somehow are contributing to the progressive MS that people have independent of relapses? And to understand which of those in fact is the case, or if it's a combination, is one of the most important things that we want to do moving forward. We will be able to understand who may benefit from this intervention. It may well be that not everybody with primary progressive MS or secondary progressive MS will benefit, but these are amongst the important questions for us moving forward.
So what's next in terms of this medication? How will people be able to access this in the future?
Well, this positive trial, of course, is extremely exciting. It's kind of a game changer in terms of our thinking of treatments that could impact people living with progressive forms of MS, primary progressive MS, and maybe secondary progressive MS. This will take some time in terms of the full data set being analyzed. The usual process takes months to a year or so until these results get presented to the regulatory agencies like the FDA in the US or the EMA in Europe in order for them to make a decision about whether this drug will come to market or not. Almost certainly, because there's such an unmet need, it will come to the market unless there's something that happens in terms of safety profiles between now and then. The results of the study, the results of the relapsing remitting study, which represent a fairly large number of people with MS who've had this treatment, suggests that the balance between the benefit and the risk is very much on the side of benefit. So one would very much hope that the regulatory agencies will see eye to eye and that this will be approved. The time at which it will come to market in Canada often lags a little bit behind the US. And of course, Canada first, Health Canada will approve, but then there's going to be an issue of each province making the decisions of how to get it into the provincial formulary. So that sometimes takes a few additional months. As a community, we will certainly push to see this happen as fast as possible.
And last question, what is the difference between this drug and rituximab? Because we have heard the word rituximab come up from time to time throughout the week. How do they differ?
So the difference between ocrelizumab, which was used in this study, and rituximab is that rituximab is what's called a chimeric antibody. So while they both target the exact same molecule called CD20, so they are both anti-CD20 antibodies, rituximab, which is the earlier version, was an antibody that was created by fusion between something from mouse and something from human. And while it seemed to work very well throughout the studies, ocrelizumab is more humanized. There's much less that is mouse. And what that may mean is not necessarily a big difference in terms of the benefit, but one of the types of side effects that people can get relates to the body recognizing the antibody as foreign and responding to it. That can cause either reactions like allergic type reactions, which again are very infrequent but conceptually could happen, but it can also have the body's immune system create antibodies against the treatment, which may limit the benefit of the treatment. And again, that happens infrequently, but there certainly is at least a theoretical advantage of ocrelizumab being more humanized as an antibody to not have those issues as much as even present at low levels of rituximab.
Thank you.
Thank you.